well as BISS also for this, because basically we teach, and I take the courses which is a very basic biotechnology techniques course. So I was just hearing a little bit of Madam what she was telling. It is wonderful. And then beforehand, I wanted to tell, I little rush because my slides were initially when the topic was given, it is agriculture, it is issues, and means agriculture biotechnology is prospect and issues sort of a thing. But I since work for quite some times, you just keep in the first slide, I'll tell. Okay, or I think I can also scroll. No, I cannot do that. I have to tell you to go to the next slide, isn't it, Megha?
Yes, sir. Yes.
Yeah. So you can go to the first slide. I'll tell that. Let me just little introduce what I'm actually going to. You go to the first one.
Yes.
You are going in the other direction. Yeah. Right. So what we actually take some basic courses on biotechnology. And each technique in detail we actually do in the wet lab. For last 1 year, I'm not able to do. So I tell all the students, if you have any doubt, you can WhatsApp me, you can mail me of any kind of thing. But today's talk, after I told that I'll be talking in nitrogen use efficiency, I was a bit scared because seeing the students' profile there, even I have seen students are from chemistry, physics, medicine are there. So it becomes little difficult for me to go to that undergraduate level and teach. I'll be more than happy to tell them, but see, 45 minutes time is not enough because I thought I'll be talking for 45 minutes. So there are some slides which you may not understand, but then the very purpose is to tell what actually we are trying to do or what way you can. And you have lots of lecture in that, and people will be going in details about the techniques, like I heard about the tissue culture, plant transformation, and do. So techniques you can use, but then where you will use and how you will use, that is very important. So here I am going to talk about nitrogen use efficiency. You can take this as a trait. It is a trait of a plant. You can do that for any trait in the sense it can be for a biotic stress, like some disease resistance, some drought tolerance, some salinity tolerant, to anything you just have a problem for a plant. You can use similar kind of approach. Exactly same will not happen for all. So basically you can say that I'm actually going to talk about nitrogen use efficiency per se, but then it is for any kind of stress. How it is an example that I have taken as a nitrogen use efficiency. So little bit of details about nitrogen use efficiency also I'll be talking. Okay, so you can go to the next slide. Basically I have, yes, please. Yeah. So basically I put it like a biotechnology and biotechnology in agriculture biotechnology, then how this biotechnology is used for crop improvement, and then nitrogen as such, how it is, and then in agriculture, what's the role of nitrogen. And nitrogen is having another big part we are actually focusing, that is nitrogen use efficiency. I'll be just telling about that also a bit. Then of that, biological nitrogen use efficiency is the part which nitrogen use efficiency. Nitrogen is not a biological material, so its use efficiency is controlled by different kind of people. I'll be talking bit of biological nitrogen use efficiency because there the plants are involved and biotechnology is going to be involved in that. And after that, of course, what are the approaches we can take? There's a slides on that. And then different biotechnological intervention and different approaches of nitrogen use efficiency. Then finally, we can go for a conclusion slide, that what actually, if you have a problem in biotechnology, how actually in agriculture, how do you? It is not necessarily that always you have to have a genetic engineering kind of a thing, because India, as well as in many countries, we have quite a few genetic engineered plant in our hand, but they are not been released. That is because of a lot of biosafety issues are there, and DBT is very well forming the rules. And with the new techniques, we are hoping that it will not. So basically, if you say biotechnology definition, this has been taken from different, one big one, then medium one, and then small one. If you just read, I'm not going to read that, but the last one, if you see, the use of biology to solve the problem and make useful products. basically the extract of that, and the Convention of Biological Diversity, that CBD, basically time to time they change this. If we just talk about this biotechnology, just hold on, yeah. So if you talk about biotechnology, what happens is we say that wine making, curd making, where we are using some biological material or some organism for making a product. Like in that case, when you talk about a product using the biological system, then even growing a plant, you are growing a banana plant, or you are growing a crop, or we are doing agriculture, everything comes under biotechnology. But in that case, everybody will say that anybody working with the living system to produce something, they will say we are doing biotechnology. Whether it is a curd making, my mother is doing curd, so I'll tell you are doing biotechnology. But then from there, it is little bit came. This biotechnology name itself was actually came much later. So why it is important is where form we say, though there is no clear-cut line that this is called biotechnology and this is not, because most of the biotechnology in agriculture, that has to be clubbed with genetics and plant. At some point of time, like when you transform, at present in India, BT is the transgenic crop which is present. But this BT is present in more than 100 varieties. So first, what it was done in one. After that, you have to do a breeding, and then you take it to a desirable variety which is a drought tolerant or kind of thing. So basically, modern biotechnology, there is little bit. You go to the next slide. Yeah. So basically, CBD again given a definition of modern biotechnology, where they're saying it is not in a natural condition. If you naturally grow something, even if whether it's a microbes or whether it's an agriculture, don't put it a biotech tag and get people confused, because it was going on. So it is basically an in vitro technique. So even for tissue culture, we call in vitro culture. We are not going in that. So that's why tissue culture came into biotechnology, though it is a plant normally growing. Even a normal plant, when you are doing a regeneration in a system, we call it is a biotechnology. And then that includes the recombinant DNA technology, means you actually cut, join kind of thing, and then you make it, and then you can use directly by injection or through a media or through a organism. You can put it in a system or in a means in a particular organelles. So many of the cases we fuse the like protoplast fusion. We fuse some of the cells and make something which is naturally not possible. So to be frank, biotechnology definition, you will get many definitions, but don't confuse. When you say it is a biotech crop or a biotech organism, and later they are telling it's a genetically modified organism or kind of thing, there it means usually in nature it does not happen. Otherwise, if you say genetic engineering, or you can say genetic gene manipulation, it has happened. Even for the evolution, the human being has come from ape to ape has come to some other organism like this. When it comes, there are a lot of changes happening in the DNA. But then basically, when we talk about the biotechnology, we usually tell something which is using the techniques which is naturally we don't do. And there is something what is called, which we are talking about, the in vitro. There a range of different molecular techniques such as gene manipulation and gene transfer, DNA typing, cloning in plants and animals. So now if you remove the animal part, we do in case of animal and plant when we are talking in vitro breeding and in vitro manipulation, but so far it has not come for human being, though human cloning also some cases some people report, but it is not there. But then it is like an in vitro kind of situation, where now combining the natural things along with the in vitro, like DNA typing. When you are talking about, basically we are talking about a marker. Like what we do, we are using biotechnology for confirming the parentage. We are using biotechnology for confirming, means for catching a criminal. We do DNA fingerprint and match it, which was not possible long time back. You know, it is not possible. And we call fingerprint because every individual will be having a separate kind of a tag. So they also fall under the purview of biotechnology. Why I told this, it is a biotechnology course, and then you must have heard many times. It is for me to keep the flow, as well as these are the things which can be used. It is not a particular technique that this only we use for solving a problem. It is an integrated approach or different kind of approach for solving a problem of a crop or even for an animal. Next slide, please. Yeah. So this is also basically what we are talking about, the modification of, when we are talking about genetic engineer or genetic modification or GM. So it is a modification of an organism. So genetic makeup. We usually change the genetic makeup deliberately. It is not naturally occurring. It is, we are knowing that why we are going to change, and because of this change, what will happen. Of course, we are anticipating that what will happen. And many of the cases, it can go beyond that. And that is the reason the biosafety is in place. That we have to check, yes, what I wanted, that only has happened. It has not actually done anything wrong. So in which DNA from one organism to other cell, we transfer without a sexual reproduction, or a gene in the host is modified. It is not necessarily that always you have to take a gene from some other and put for telling that a genetically modified. You are having a gene of yours, means a plant, and then we wanted to actually transfer, we wanted to change that, and then its character itself will get changed. Next slide. Yeah. So now coming to the agriculture biotechnology, so far what I told, exactly those things has been put together. And then I have given an example from USDA. We have different discipline, even different organization is giving that terminology. So it includes the traditional breeding also. Like as I was telling, no, when we are talking about a BT cotton, it is a biotech crop. But unless and until breeding is included into that, it will be just confined to one particular plant where, or one particular plant type where you have transferred. After that, we are not going to do any kind of transformation. But then what we do, we actually cross that with a desirable variety and do that. So other than that, everything what we actually doing, means whatever I have told, that will be that is included. So it is like a living organism, a part of the organism, make or modify the product. This is to improve the plants or animals. So basically, this is what in case of agriculture biotechnology. So I think it is good enough so far what we told. Next. Now, when we go for application, these are the different places we talk. Just last I was talking, somebody was asking about the vaccines. Madam has given the answers, and it is in place. It is going on. But there are a lot of things. Overall, we can produce some product, like even you can do a metabolites produced to plant. Means you can improve the plant. That is one part. Or you wanted to target a product by a plant. There also you can actually do that. Next slide. Yeah, so this is a timeline. I'm just skipping that. You can have, since you are the students, this how it actually happened. So entire thing, if we talk, this can be told as biotechnology, that from how they have domesticated to how actually we came to this bioinformatics and genomics. Next. Yes. So basically, in that biotechnology, the students who are reading, they read molecular biology and biotechnology. In fact, our discipline's name is also MBB, molecular biology and biotechnology. Molecular biology is the science part. You are knowing deep into the science how it is happening. It is a Okazaki fragment. It is your replication. It is your what is called specifically how unwinding is happening, to everything we know about the molecular biology. But then, as long as those knowledge is not being used, they are in the science itself. So there is the difference between science and technology. And when a technology people, you get a BTech biotechnology. I actually am quite confused with the students who come, BSc biotechnology, MSc biotechnology, BTech biotechnology, MTech biotechnology. And ultimately, they did almost similar kind of thing, because a biotechnology students cannot go without knowing molecular biology. You have to know what is happening, then how you are applying it. So both the cases, it is quite clubbed. Because when we are talking, the subject itself is a technology. How can you tell it is BSc biotechnology? It has to be a technology. So ultimately you start with a molecular biology and then you come with that. The second part I have already told, conventional agriculture versus biotechnology. Conventional agriculture is normally you are just developing a variety by crossing and then selecting them. And then you are telling, yes, this variety is very good. But when you are coming to the biotechnology, which we naturally don't do, we use the molecular biology techniques, and we know if I actually manipulate in such way, and then what will happen is we'll get such kind of product. So there it is. And then what we have already told about the modern biotechnology, already CBD has defined that it is basically a DNA based technology. When it comes to our nucleic acid based technology, it's not always DNA, it is nucleic acid based technology. There it is. But don't confuse, sir, I am drawing on protein, whether it is molecular biology or not. It is molecular biology. Proteins are much smaller molecules. Proteins has a lot of control, and they are all actually coming from, since the genetic material is DNA, that's what we are telling about that. But there is a clubbing of all. Next slide. So coming to the nitrogen. So I now come to that part. We were so much worried about oxygen for some time, but in the air, if we remove the small part of there are a lot of elements present in the atmosphere. Of that, carbon dioxide is one of the important part, because because of the carbon dioxide, plant photosynthesize and we survive. But then similar is in case of nitrogen. Oxygen is very much required for us, which you say, if you say it is 20%, then 80% is nitrogen, 78 precisely, if we are saying. Its atomic number is 7, mass number is 14 point something. So this is what is almost like carbon as its carbon dioxide as it is there. Carbon dioxide to some extent can react, but mostly nitrogen, whatever is there in the atmosphere, is quite inert. Many of the cases, we actually use nitrogen gas to make the things inert. Like many of the cases, packing we do know. In the packing, if you are just leaving it, then there will be some kind of some oxygen or some gases will be there which will react and they will become rancid or something. So they usually purge with nitrogen. So they actually push the nitrogen so that nitrogen is inert. So basically it is very inert. That nitrogen has to be brought inside the body as an organic material, and that process is called assimilate. Like as photosynthesis is called carbon assimilate, similarly nitrogen has to be assimilated. Unfortunately, as we cannot do carbon assimilation, we cannot do nitrogen assimilation also. We have to actually depend on microbes, or basically it is on microbes. And some of the cases we call it is plant, but it is plant who does not do, it is done by the microbes. So next slide, please. Hello, next slide. It's actually not moving. Hello. Hello.
Sir, just a minute. I have some internet issue.
Okay. Shall I try once? Just let me see. Let me see. Share screen I can do. No, this is desktop. Yeah, just a minute. Yeah. Are you able to see my? Are you able to see my screen?
Screen is visible, sir, but your presentation is not.
Yeah. I'll open this presentation. Don't worry. This is the one. Yeah, it's okay.
Yes.
So just hold on, because next slide is also becoming a little bit problem. So fine.
Yes, sir. Perfect.
Okay. So now coming to this, basically I am talking about nitrogen, and all of us has studied, whether we are in science or arts, because up to matriculation we all had to study science, and in that, basically the nitrogen cycle, all of us know. Here the numbers what they are telling, it is basically teragram per year of things what is happening with the nitrogen. Teragram is 10 to the power of 9 kg. So if you see there, this is Haber-Bosch process is nothing but fixing the nitrogen chemically in a factory, and the fertilizer is made, and those fertilizers are put there. This is a huge amount, and you can say among all it is 120. Other than that, there are a lot of other things. Just can you just excuse me for a minute? One second. Just give me 1 second. Hello, able to hear?
Yes, sir.
So actually what I was talking about this slide. So actually in a nitrogen cycle, we fix the nitrogen. Other than that, the chemical process, lot of nitrogen is fixed by different organisms. And then after the fixing, they get after the fixing usually happens in the form of ammonia, then it becomes nitrate, and then again it becomes denitrified, and this nitrate again become denitrified, and then it again goes to the atmosphere in different forms. So basically for the plant nutrients, when we are talking about the plant nutrient per se, the nitrogen is the maximum amount it is required. Carbon, hydrogen, nitrogen, carbon, hydrogen, oxygen, it comes from the water as well as carbon dioxide from the atmosphere. But when you are talking about the other nutrient, macronutrient, which we are applying as a nutrients in the plant, there are lots, there are many. So as I told, micronutrients are there are macronutrients, but we are concerned about the nitrogen, which is maximum required for the plant and given in the form of a fertilizer. And if you are talking about the nitrogen, because we are talking about the nitrogen use efficiency, I'll come to the definition. But if you see this left side and right side, almost similar. That means this is actually nitrogen fertilizer production. Over the years, how the nitrogen fertilizer is being produced, and how fertilizers are produced means they are produced for the sake of utilization, and that is the reason it is almost same way the amount it is produced. These are all FAO's data, and then they are being used. And then in this case, it is a typically 2014 one graph I just showed, different parts of the world how they use. The thing from there we wanted to say is nitrogen is being used in a huge amount for agriculture purpose, and what it does. So what it does is, see, if we are using 100% nitrogen, of that only 50% is being used, and rest of the 50, I'm talking with a big optimistic figure. 50% if the plant is able to use it, or the plant is taking it, use will come to later. But then if the plant itself is able to take the 50% of the nitrogen what we are using, that is a great achievement. Here it is a BASF is given because of a purpose, it is BASF. And since I took the slides from the BASF site, I did not remove their name. So the very purpose is to tell, okay, just 1 second. So here what we wanted to show was, like when we are putting there the nitrogen, this atmospheric nitrogen is coming into that through the industrial processes, fertilizers and all, then plant is taking, there are a lot of amount which actually goes and gets lost. And then as I told, it is ammonia. From the nitrification process it becomes nitrate, and then in a denitrification process it gets lost. and this orange color and this orange color, these are losses. So in that loss also, they have given the distribution, how the loss is. But before that, forget about other three. On the top, what we are talking to, what we are trying to tell, it was nitrogen use efficiency in 1960 was, in case of top graph is India, it was high. That means if we are using little bit of nitrogen, we are getting the yield was much better. But the days and days it is coming, we are keep on using more and more and more nitrogen, and that way we are not getting the yield, rather it is getting loss. And that is how the nitrogen use efficiency, the top graph you are seeing, with the year it is gradually and gradually slowing down. And this is a similar kind of graph. If we say in this, what is happening is this top one, if you say, you forget about all this. Here we wanted to say which crop, how much it is taking. Of that, if you see the cereal, two cereals, no, rice and wheat, they take the maximum amount. And this left side graph, what we wanted to say was, this is nitrogen, whereas phosphorus and potassium use has not increased so much. Our production also has increased, but then, you know, the nitrogen is going sharp high. People are trying that if we use more and more urea, we'll be getting more and more crop, but it is not so. And the food grain, so naturally, if we are using the same amount of nitrogen, you are expecting that food grain will produce more. It is not happening proportionally. And the last part of right side, what we wanted to say, if we talk about the extreme unfavorable condition, there the nitrogen use efficiency is only about 20%, mean 80% of the nitrogen get lost. And when everything goes favorable, there we get, at present, our aim is to at least 50% we should be able to use it. Now, what happens in case of in soil? There are two types of fertilizer usually we use, ammonium nitrate and urea. Ammonium nitrate is mostly, it is banned now, and then we don't use it, except for the lab experiment we don't use ammonium nitrate, but we use urea. So urea goes to the soil, and then with the enzyme called urease, it becomes ammonium carbamate, then it becomes ammonium carbonate, and then that is basically ammonium ion. And that ammonia, in the process of nitrification, it's become nitrate. That nitrate is taken by most of the plant. Of course, some of the ammonia, some amount of ammonia is taken by rice plant. Rice takes mostly ammonia. Of course, it can take nitrate also, but the other plants, it takes nitrate. But this ammonia get lost, and again this nitrate get leached down to the soil, and it runs off and goes to the ocean, to pond, to different places by running off. And then this nitrate becomes denitrified, and it become, we call it as NOx, means that becomes very, very obnoxious kind of gas, which actually harms the atmosphere. This is what they have distributed, that 80% loss applied can be lost as ammonia to the air, 3% during nitrification process. This is another 20% during nitrification, up to 20% applied nitrogen fertilizer. So this is just a data, but forget about that. It is not that necessary. Left side, what we are talking about is the nitrogen what we have given. This is what is the fate. One is volatilization, it is going, denitrification process. Another is runoff. It is just going and going to the river, pond, to that. It goes to the underground water and it pollutes the water. Some part, which our target at present, we are trying for 50% or 60% maybe plant use. And this green part is our area of work. There are two slides. What we wanted to say is, the nitrogen, when as long as it is there in the atmosphere, it is actually not reactive. But then when it forms ammonia and different nitrate and different gases, they are called reactive nitrogen, and they are quite harmful. And those reactive nitrogen of that, if you say NO, NH3 and N2O, these are the gases which actually form the first, second and third most important greenhouse gas. So the contribution of nitrate pollution or nitrogen pollution is much more harmful than even carbon dioxide for a greenhouse gas or global warming it is. So And this is what it is telling. N2O is almost 300 times greater than carbon dioxide for forcing the climate, for climate change. So now coming to that, what I was trying to tell is nitrogen use efficiency may not be, you all biotechnologists are so much keenly looking into that, but it is a very, very important aspect. Government of India at present, our Prime Minister has told that any kind of urea is sold, it has to be neem coated. Why it is, we'll come to that. So what is the purpose of doing such kind of research? We wanted to actually reduce the loss of nitrogen. How much we are using, that should not be lost, or in the other way, whatever actually we are trying to apply, that much should be useful. Or you can say in the means in a different way, that as much is required for the plant, that much only we should be using. For that, we have to increase the nitrogen use efficiency of the plant, means by definition. So what is the definition? Output by input of nitrogen. And now the output in case of a cereal crops, because you have seen rice and wheat, which actually takes the maximum amount of nitrogen. So output by input meaning is how much of grain you are getting, how much of yield you are getting per unit of nitrogen, per kg of nitrogen applied. So this is what is actually the purpose of doing this research, that we should be able to use less amount of nitrogen, but then suppose we are not using anything, even then we'll get some yield. But that yield should not be so less that we will not be able to feed the entire population. So when you are talking about reducing the nitrogen, naturally, if you are reducing the nitrogen, some amount of yield loss may be there, but it has to be compromised between two. That you will be reducing the nitrogen in such a way so that the nitrogen, the yield loss should not be that substantial. Now, if we are talking about the nitrogen use efficiency, before coming to the biotechnology, there are some management aspect also. Since we are talking about the agriculture and nitrogen, I'll just touch on that. And then there are other two methods. One is by biological nitrogen fixation and enhancing the nitrogen use efficiency of the plant. So these are the three aspects of it. So management, how do we do? As I told, no, this is urea, urease, and then there are nitrification. In this nitrification, there are some bacteria, which is Nitrosomonas and Nitrobacter category of bacteria. There are many, so which actually forms the nitrate. And this ammonia and nitrate both are useful for the plant. So if we are able to reduce this process, it has to be there. But in this urease, suppose you are inhibiting, you are applying some inhibitor to urease, or you are applying some inhibitor for nitrification, then you can actually, the urea what you are giving, that urea, if anybody has used or seen, you will see that it actually gets solubilized instantly. And if it is solubilizing, after that, it slowly and slowly by urease, it becomes ammonia, and then it starts evaporating nitrate and starts leaching. So there are a lot of chemicals. That is how this BASF came into picture. They are selling chemicals, what they say, urease inhibitor and nitrification inhibitor. If you are applying the inhibitor, they actually give a calculation how much you can solve and all. But they are again chemicals. They have a side effect which they hide many of the cases. So that is the reason we are actually recommending that neem coated urea. If you are coating the urea with neem oil, the neem oil is having a urease activity. It is organic. At the same time, it actually slowly, the urease will be active and it will be forming the ammonia and nitrate. Other than that, through management, there is an application schedule. The fertilizer, morning itself, if the food is given to a person that you have the whole day food and go cannot. You have to split go, because the efficiency will be better. So same way, initially the fertilizer used to be added in a same go. At one time, the nitrogen will be given. That's all you forget. But now the stage has come after two doses, 3 doses, 4 doses. Now they are telling daily with the fertilizer, with the irrigation, you put a drip irrigation, as much water as the nitrogen or nutrient is a resource, water is also a resource. We are working on water also. So water is very, very important. So when you are going to use the water, you should be used very judiciously, and along with the water, you can apply the fertilizer. So this is the management part. Now the next part is coming, biological nitrogen fixation. Here, mostly the bacteria are involved. There are some free living bacteria like a cyanobacteria, azotobacter, which fixes their nitrogen. And there are some plants which host bacteria in their roots. Okay, so they host the bacteria. Those bacteria actually, what they do, they will fix the nitrogen and give the ammonia to the plant, and the plant will give the carbohydrate. This is the understanding. And you know, most of the cases that leguminous plant or symbiotic organisms are present there, like a rhizobium. Now, in this, what we can do in biotechnology, we can actually improve the efficiency of the nitrogen. Means efficiency of their fixation. How efficient those microbial strains are. So those who can fix more nitrogen, we just select them, then we use them as a biofertilizer. That's one part. And then we can genetically modify them. Genetically modify them in a different way so that their efficiency gets improved. And third is, you actually try to incorporate the genes inside the plant itself, like rice, if you can do that. This is quite challenging. Like if you say we wanted to put the gene in some animal or even us, and then we'll do photosynthesis kind of a thing, but still we are working on that and we are quite successful. In our lab itself, we have a project on biological nitrogen fixation. There we are trying to make the rice plant which can actually fix nitrogen like legume. I'll come how we are doing into that. So this is one of our paper. It has been published in one of the journals in that. This is this applied and environmental microbiology. The lead worker in that was the head of biotechnology, Dr. H.K. Das. He was the head in JNU. So that part is genetically modified. You forget all this intricacy. Here, this concentrate on the NIF-L part. NIF-L is the portion which controls the nitrogen fixation. When the nitrogen fixation is beyond a limit, in the sense, if the nitrogen fixation goes a little more, then they say you stop it. So in this process, this portion has been deleted. So that organism or that bacteria, it is in azotobacter, that bacteria could not sense at all that he has enough nitrogen fixed. And it is successfully done, and we could reduce almost 40 kg, 120 kg is the recommended urea for recommended nitrogen for wheat. In case of wheat, when you are applying that genetically modified organism, we are seeing that we are able to save 40 kg. That has been done. That is in DBT RCGM trial, it was successful. But then, you know, it is releasing is a different altogether aspect. So this is one example where we can genetically modify and improve the biological nitrogen fixation. Now, the other part is what I told we wanted to do, and there's a big project, almost 6 years running this. This is biological nitrogen fixation in rice specifically. So legume hosts the bacteria. There, what it does, legume hosts bacteria, and by forming the nodule. Nodule is, you are making a house for the bacteria who come and stay here. I'll give all the facilities. You fix nitrogen. That's what it does. And bacteria fix the nitrogen. So that is what happens in case of a nitrogen. So we have two strategies. Second strategy we have started this year, but the first strategy we have already quite successful. There we are transferring the genes for nodule formation from the legume to the rice plant. So you take one by one, like as madam was telling, you do the transformation, and one transformation we do, two transformation we do, three transformation we do. Then we cross those plant like by plant breeding, and we bring together. And then we are seeing, yes, now we are able to see in microscope that yes, we are able to actually form a sort of a nodule kind of a structure in that. It is long way to go. So if we are giving a congenial atmosphere for the rice plant root to form the nodule where the rhizobium will come, in the soil rhizobium is there, even inside the plant rhizobium is there. but then they are not able to form the colony, they are not able to fix, and because they don't have nodule. So we are transferring the gene to form the nodule in rice. The other strategy is, instead of doing that, why don't we do the nitrogen fixing gene? Means NIF gene itself we wanted to transfer. Around 12 to 13 genes has to be transferred to the plant. But this is a challenging thing because nitrogen fixation happens in complete anaerobic condition. So we have to transfer to a organelles where it is oxygen is depleting. So mitochondria is one of the things, but still we are working on this. So this is one of the things which is very interesting. Now coming to the plant. Among the plants also, you will be seeing our wheat. You give less nitrogen, they produce good yield. And there are some plants which are actually not able to, we are giving too much of nitrogen, but in spite of that, yield is not much. That is because there are lots of gene present in the plant. So actually this nitrogen use efficiency is depending on a plant. This is the third aspect, and where we are again working on that, how much plant can take. And whatever plant has taken, soil it is there, it is not necessarily that plant will be able to take. So that is called uptake. There are lots of proteins and lots of gene involved in uptake. And once you have taken, how you are assimilating. As I was telling, no, carbon fixation will happen. They assimilate to the amino acid. So once the assimilation has happened, that means they have imbibed. The atmospheric nitrogen has become nitrate, and then that nitrogen is taken by the wheat plant, maize plant, and then they have assimilated that, and then they have become amino acid, and it has come into the body. But not necessarily that is always useful, because if your body is good, like you get lots of biomass. Your plant is very, very good looking, but your yield is very less. So that's also not going to be useful. So how good your utilizer, that is also, utilizer means how good you are able to give the yield. So that is what the three aspects which we are working. So what we are trying to see, we want to take a plant which is very poor in nitrogen use efficiency. We take a plant which is very good in nitrogen use efficiency, and then we give a nitrogen stress. And after giving the nitrogen stress, we actually take the RNA from there, because we see which are the gene actually getting expressed because of giving stress in an efficient genotype. And then we find out what are the gene responsible in that. And if we are able to find out the gene, then it is very good. But this kind of characters, they are called abiotic stress characters. No? So these kind of characters are not controlled by a single gene. Usually you will not get a single gene which will tell yes because of that. So what we get? We get a portion of the chromosome. If that chromosome is present, then we are seeing yes, they are. And then again, we are seeing there is another portion, maybe 4 different portion in 4 different chromosomes, it is present. And if they are present, then these plants are, then in that case, these plants are very, very efficient. So these are the genetic potential of the plant, and all those regions of this chromosomes, we call it as quantitative trait loci. They are not a single gene is working. These traits are quantitatively decided by different loci. Loci means a location of the chromosome. So all these strategies together, we are trying to work on that. And this is an uptake, how it goes. If you give the fertilizer with the time, how it goes. As I told, some of the slides I'll skip. And then for the uptake, root is very, very important, and different root in that. This picture shows the different root is having a different role in uptake. And this slide shows that lots of transporter. Some of the transporter we call high affinity and low affinity. When the nitrogen is very less, this transporter comes into the picture. They are called high affinity transporter. They have affinity is very high, and low affinity transporter, when you have enough nitrogen, this high affinity will not work. There are some which are dual affinity, which work both the cases, and then they transfer into the root. From there, there are a lot of transporter, even up to the seed, they transport this nitrogen. And those transporters are nothing but protein. So since they are protein, they are controlled by gene. You can have those, and then you can manipulate them for things. So primarily the incorporation of the acquired nitrogen in the biomolecule of the plant, and ultimately using this biomolecule efficiently to produce the economic yield. Taking is not all. Synthesizing to a proteins and making your body or biomass is not all. You have to reflect that in an economic yield. Okay. So in this case, basically when you are talking about the assimilate, glutamate is the assimilate. And this glutamate is involved in everything, for chlorophyll, for nucleic acid, for any kind of things. This glutamate is only the first amino acid which produces, and from there everything happens. And this is the things what I am trying to show is how it goes. And there are quite a few genes which is actually involved in that, right? And this is the process, how you do this, how you acquisition of the nitrogen from the root, and then how you assimilate, means how it goes to the body, and then how you utilized for making the COP. Here it is economic yield. And remobilization is another important part. The proteins which are formed in the body, many of the cases, the older leaves will dry, dry off. So when it dries off, it does not throw this. It actually, what it does, it actually pass on that. That's called remobilization. And photorespiration is one of the process, in the process of photorespiration also, this actually recycling of this nitrogen happens. And these are the kind of genes, mainly candidate genes. We know them very, very well. And people try to manipulate them too much. There is not much of success. So we are, and this is the process, this is nitrate, nitrite, ammonia, and then glutamate, it is formed. But when you are talking about the amino acid, this most of you will be knowing, whoever is there in the biological science, that in the amino acid, it is not the ammonia alone. We need a carbon chain. And for that carbon chain, we actually require this, all our Krebs cycles intermediate. And alpha ketoglutarate or 2-oxoglutarate, this is the molecule. So there are regulators which says, okay, you give ammonia, I'll give you a carbon chain. So together we'll produce glutamate. That is what actually it produces. And as I was telling you, demobilization in case of senescence and photodespirations is how good you can recycle. My nitrogen should not be waste. My last leaf is getting dried, so I'll give it, and how good you can do that. Okay. But as I was telling, now we are more concentrating, since the candidate genes are not working, we are more concentrating on the regulation part. So there are a lot of transcription factors. There are transcription factors, means in case of gene expression, they have a role. They have sometimes negative role, they have sometimes positive role. And then there are some proteins, specifically they are actually PI2 proteins for nitrogen, AP1 protein for nitrogen, sugar transport protein for nitrogen. They are all nitrogen related specific proteins which actually controls. Okay. And then our last one is microRNA. This slide only shows that there are so many microRNAs, and one microRNA had lots of targets, right? So this is just an example of how microRNA works. So now this is almost, I am coming to the end. Approaches is, we can do conventional breeding. You take a good variety, and a good variety means efficient variety. You just cross it without knowing anything. You select whatever good is coming, and then yield also is giving, as well as nitrogen is also required less. By that way, you can develop a variety. Then, as I was telling, that if you have found out a gene or a part of the chromosome, which is called QTL, you can make a marker. Marker means you actually design a primer from that region, and you can track. You are doing a conventional breeding, but during the breeding, you don't have to wait till the phenotype comes, because yes, my gene is already present. I have already seen when the plants are very small, or even take a few seeds and do that. And for that, you actually have to do the identification of gene. How do you identify the gene? One example I gave, you give a stress, and then you actually do a transcriptome analysis. This is basically gene expression study. Or you can do a random mutagenesis by tDNA or something. You are mutating the plants in different places. So many places you are doing mutations. And then you are finding out where all mutations have happened. And then you are seeing that which mutation is giving you a better result. That's called validation. So for that, what you have to do is, if you have identified a gene, that gene has to be overexpressed in a plant. And then after overexpressing, you have to confirm that, yes, I have overexpressed, and then see my nitrogen use efficiency is increased. So that is through a transgenic approach. The last one, which we are working now, and it is quite exciting, and maybe our DBT also relax little bit of regulatory aspect, that is by knocking down. Earlier they were doing by RNAi, they had a lot of off-target effect. I'm not going into details of how it is RNAi, that's a different kind of a class. But then now we are doing through CRISPR-Cas or genome editing. Of that, CRISPR-Cas is most prominent. So if you have some genes which are hindering, which are very bad, like they are actually negative regulators, what you can do is, I am giving an example because I have already started working on this BT2. This is a gene family which is known. It is known in Arabidopsis, it is known in rice. Wheat, it is not known, but we are thinking that wheat also it will be similar kind of a negative effect. If this gene is expressing, then nitrogen use efficiency, your transport itself is reducing. Means taking up itself is reducing. So what we wanted to do is, we just wanted to do through a CRISPR-Cas. And CRISPR-Cas is such a beautiful technique. By one generation itself, you can make that gene free. Means there won't be any transgene. Your gene will be mutated. Means wherever you want, your target place will be mutated or it will be inactivated. You can do many things with CRISPR-Cas, but I am telling you, you can knock down. You can completely eliminate that bad effect of that. And that is what we are doing all the things, but latest is this. And since there is no transgene, we are anticipating that there be regulation will be not so much strict, and we'll be getting some variety on. So my lecture part is over. This is what the field looks. When you are working on a nitrogen, it is a big challenge, because this field took almost 4 to 5 years. This is third year's picture, but at present 4 or 5 years where you can't give nitrogen at all. And then when you grow the crop in the field, it looks like this. Here, same variety, same plant. Here it looks so green, and this looks like that. So this kind of field has to be developed. And many of the cases, we make a trench here, and then we make a boundary wall, so that nitrogen is very much mobile, labile. So it passes from one to another. Actually, this is not the same picture. These are 3 replications, 3 different places you have to do this. And then we do in a pots also. Pot also we do, because field, many of the cases, it is beyond our control. Nitrogen fixation happens, lot of nitrogen fixation happens through rainwater. And we don't have control in the field for rainwater. So you are not giving nitrogen, but your God is giving nitrogen to the plants. So you can't. But in this case, suppose you are putting a shade on the top, or you are putting in a glass house, or you are putting in your culture room. So you can completely do. You can do in a control media, you can do in soil, you can do in hydroponics. So that's how it goes. And this slide shows that in nitrogen use efficiency, this Arcadia is one of the company. They claim that we have gene. Basically, they have done alanine aminotransferase. This is one of the gene. They say any alanine aminotransferase, if you overexpress is any of the plants, then it will give you nitrogen use efficiency. So far, no product. Our Maheko has purchased that in India. I tried contacting. So far, there is no product or no variety came into picture, but they have lots and almost on 15, 16 patents on this. And take home message is that the tools are available. Tools are available for biotechnology. It is how beautifully you can think, how beautifully you can engineer your construct so that it will be giving you, in case of CRISPR-Cas, we are doing 6 guide RNA, means 6 gene at a time. We can, in one construct, we can actually eliminate them. So tools are all available there. Most important is you have to understand the trait. Being a biotechnology student, our major drawback is we don't know the subject much. Subject in the sense, I'm not talking about the techniques, but the problem. When you are going to solve a problem of an insect, you have to be an entomologist. When you have to solve a problem of a disease for a plant, you have to become a pathologist. When you wanted to solve a problem of, suppose, means yield, then you have to know agronomy. So we know the techniques, but we don't know the depth of that. So understanding a trait is very important. Last 10 years, I understand little bit of, because last 10 years I'm working on nitrogen. So I understood little bit of nitrogen. Biotechnology, since we are studying for last 35 years, we knew day by day it is developing. But how beautifully you can use it is very important. Then you have to identify the genes. Like there are different ways of identifying the genes. And then you have to identify the regulators also. Then manipulate as per your wisdom. So you can do that. So that's all. Thank you. I don't know whether you have time, and you can ask as many questions you can. There's no problem.
So students, quickly, you can ask your questions if you have. we'll be starting our next session by 12:20. So please go ahead with your queries. Any question, anything about agriculture biotechnology?
Anything, yeah.